All right, science enthusiasts, buckle up. You know that moment when you're about to tackle a hands-on lab experiment, and you don't just want to follow the steps blindly. You want to truly understand the fundamental science at play, to feel completely prepared and confident before you even pick up a test tube. Well, you've hit the sweet spot. Today, we're giving you that essential shortcut to being well-informed, a deep dive into the very bedrock of life. Biological molecules. This isn't just about mixing chemicals. It's, about unlocking the secrets of what makes up everything from your cells to the food you eat.
And what's truly remarkable here is how foundational this knowledge is. Seriously fundamental. We're going to unearth the core concepts that underpin Lab 3, biological molecules, equipping you with the insights you need before you even set foot in the lab. We'll cover the vital how-to of the procedures, highlight the critical materials, and even arm you with some ingenious strategies for those pre-lab quiz questions. Our mission today is to ensure you're not just ready for this lab, but genuinely confident and maybe even, a little excited for what's ahead.
Absolutely. Think of this as your personal guide to cracking the code of carbohydrates, proteins, and lipids, transforming what might feel like a complex set of instructions into a clear, fascinating journey of discovery. Let's get started and demystify the building blocks of life. Okay, let's unpack this with the big question first. When we talk about biological molecules, what exactly are we referring to? And, why are these particular molecules so incredibly crucial for life as we know it?
It's a great place to begin because if we connect this to the bigger picture, well, all living organisms on Earth, the simplest single-celled bacteria to us complex multicellular animals, are fundamentally built from just four main classes of organic molecules. You've got carbohydrates, proteins, lipids, and nucleic acids.
The big four.
Exactly. And these aren't just random components. Each class is assembled from characteristic smaller units or subunits that link together to form much larger, more intricate structures.
Okay.
To really break that down, we use two key terms. First, monomers. Think of these as the individual single-unit building blocks like individual Lego bricks,
Yeah, like Legos. Got it.
Then, when you connect these individual bricks together, often hundreds or thousands of them, you form much larger, more complex structures we call macromolecules. These are essentially large, complex polymers built from those linked monomers. It's the elegance of simple units creating incredible complexity.
Simple units, complex results. Makes sense.
Let's see how this applies to our main players for today. For carbohydrates, the monomers are called monosaccharides. A fantastic example, and one you're probably very familiar with, is glucose, that simple sugar our body uses as its primary fuel source.
Right, glucose.
When many monosaccharides link up, they form larger polysaccharides, things like the starch you find in potatoes or rice, which plants use for energy storage, or the cellulose that gives plant cell walls their rigidity.
Starch and cellulose, okay.
Then we have proteins. Their essential monomers are amino acids. These amino acids link together to form long chains called polypeptides, which then intricately fold into our functional proteins. And these proteins are the true workhorses of our cells, responsible for pretty much everything, from catalyzing reactions as enzymes to providing structure in your hair and nails.
Workhorses, I like that. So amino acids make proteins.
Exactly. Now, lipids are a bit different. They don't always follow that strict repeating monomer-polymer chain rule in the same way carbs and proteins do. They are generally composed of smaller units like glycerol and fatty acids, which come together to form larger fats and oils crucial for energy storage and cell membranes.
So lipids are the odd one out, structure-wise.
A little bit, yeah. And finally, though our lab today focuses on the first three, we can't forget nucleic acids. Their monomers are nucleotides, which link to form DNA and RNA, the very blueprints of life itself. But for this deep dive and for Lab 3, our focus is squarely on carbohydrates, proteins, and lipids.
Got it. Carbs, proteins, lipids.
So how are these incredibly complex functional macromolecules actually built from their simpler parts? This is where a fundamental process called dehydration synthesis comes in. It's a key chemical reaction that's constantly happening in all living systems, forming the very fabric of life.
Dehydration synthesis. Removing water to build.
Precisely. Imagine two of those monomer units sitting side by side. When they're ready to join and form a larger chain, a remarkable little chemical trick occurs. A hydroxyl group, which is an OO, is removed from one monomer,
Okay.
and a hydrogen ion, an OU, is removed from the other. Now, if you combine an OU and an OO, what do you get?
HO. Water.
Exactly. This removal of water, which is why we call it dehydration, is exactly what allows a covalent bond to form between the two monomers, linking them together. So dehydration synthesis literally means building by removing water. This elegant mechanism is how living organisms construct all those intricate, complex biological structures they need to survive, thrive, and reproduce. And here is a mind-bending thought. Your body builds millions of these bonds every second. It's an energy-intensive, constant construction project happening inside you right now, all powered by this simple yet profound trick of removing water.
Millions per second. That's incredible. Forming the very fabric of life.
Yeah.
That's a powerful image. It's amazing to think all this complexity starts with these tiny molecular Lego bricks. So it's not just about what they are, but how they link together through this clever water removal that creates such diversity. But once they're built, how do we actually identify these molecules? How do we spot the carbs, proteins, or fats in, say, a food sample? This leads us right into biochemical testing, doesn't it?
It absolutely does. In any scientific investigation, especially when using chemical indicators, ensuring reliable results is paramount. This is where the concept of controls becomes indispensable. Knowledge is most valuable when understood and applied, right? And controls are kind of the unsung heroes that validate our findings, giving us rock-solid confidence in what we observe.
Unsung heroes, I like that.
Many biochemical tests, like the ones we'll dive into today, rely on specialized chemicals called indicators. These indicators perform a really neat function. They change color in a specific, predictable way when a particular target substance is present. This color change provides a crystal clear visual cue, a signal telling us something important is happening or, well, isn't.
A visual signal, like a chemical traffic light.
Kind of, yeah. But a color change alone in isolation isn't enough to draw firm conclusions. We need context. And that's precisely why controls are essential. Controls are samples that are explicitly designed to show both the positive and the negative results for each test. Think of them as your scientific benchmarks, your gold standards.
Benchmarks, okay.
They help you evaluate the accuracy of your own results and, crucially, verify that your experiment actually worked properly from start to finish. Without them, you might see a color change in your unknown sample and not be totally sure if it really means your molecule is there or if it's just, I don't know, a quirk of the reagent or your setup. They act as an unwavering standard for comparison, which is absolutely critical for making accurate observations and drawing valid conclusions.
So they keep you honest, basically.
Pretty much. And we specifically use two main types of controls to achieve this clarity. First, there's the negative control. This is a substance that, with absolute certainty will not react in the test.
Like plain water.
Exactly. Its purpose is twofold. It serves as a baseline, showing you there's no contamination in your reagents or equipment that could give you a false positive. In other words, it confirms that a positive result isn't just a false alarm. A classic example is using distilled water, which we'll often call DW in any of these tests. It should never produce a positive reaction. If your distilled water tube does change color, you immediately know something is fundamentally wrong.
Okay, red flag if the water changes color. Got it.
Then, on the flip side, we have the positive control. This is a substance that you know will give a clear, unmistakable, and robust reaction in the test. Its purpose is equally vital. It confirms that your reagents are fresh and active, that your equipment is working, and that your technique is correct.
So it proves the test can work.
Precisely. If your positive control, which should show a clear reaction, doesn't, well, then you know something is fundamentally amiss with your experiment. You need to troubleshoot before you can trust any results from your unknowns. For instance, a known glucose solution used in the Benedict's test should always show a clear positive result. If it doesn't, maybe your reagent is old or your water bath isn't hot enough.
That's a crucial point. It's like having a confirmed no from the negative control and a confirmed yes from the positive control to really understand what a maybe means in your unknown sample.
Yeah.
Without them, you're just guessing at what those color changes signify. Huge room for error. So with these indispensable benchmarks in mind, how do they guide us through each specific test? Ready to zoom in?
Let's do it. We'll be focusing on identifying 4 primary types: reducing sugars, starch, proteins, and lipids. Each test has a unique chemical mechanism, and understanding that mechanism, not just memorizing the color, is really key.
Okay, let's start our molecular detective work with sugars. What makes a sugar a reducing sugar anyway? And how does the Benedict's test work its magic?
Good question. The key insight here is the chemistry. Reducing sugars possess a specific functional group, usually an aldehyde or ketone group. This includes a double-bonded oxygen atom, that CO group. Think of that CO as a molecular handle that lets these sugars readily donate electrons.
Donate electrons, so
Yeah.
they reduce something else.
Exactly. They act as reducing agents. This specific functional group is precisely what reacts with the copper ions in the alkaline Benedict's reagent.
Ah, yeah?
So the core principle of the Benedict's test is an oxidation-reduction reaction, or redox for short. When heated, the reducing sugar gets oxidized, it loses electrons. Simultaneously, the copper ions, specifically the blue copper ions, Cu in the reagent, get reduced. They gain electrons and transform into copper ions, Cu. These Cu ions are insoluble and form a colored precipitate. That's the color change you see.
So the sugar gives electrons to the copper, changing its color.
Spot on. The reagent is Benedict's reagent itself, initially that clear blue solution. When you heat it in a boiling water bath, and the heating is crucial, the reaction and expected results are quite striking. The color changes in a progression related to the amount of reducing sugar present. If there's none, it stays clear blue. But with increasing concentrations, you'll see blue, green, yellow, orange, and finally, a brick red precipitate.
A whole spectrum, so it's not just yes, no, but how much.
Pretty much, yeah. It gives you a semi-quantitative idea. Now, what it specifically detects is important. All monosaccharides, like glucose and fructose, are reducing sugars. Many disaccharides, like maltose and lactose, are too. But crucially, sucrose, common table sugar, is not a reducing sugar in this test. That's key for later.
Sucrose is a no-go for Benedict's. Got it.
Right. Now, for the controls for Benedict's, you'll set up 2 test tubes. Tube #1 gets glucose solution, that's your positive control. Tube #2 gets distilled water, your negative control. Add Benedict's reagent to both tubes. Swirl gently to mix. Then, absolutely critical, place both tubes into a boiling water bath for 5 minutes. After heating, carefully remove them, maybe let them cool a bit, and observe the colors.
So positive controlled glucose should change color. Negative water should stay blue.
Exactly. A positive Benedict's test is any color change from clear blue to green, yellow, orange, or red precipitate. A negative Benedict's test means the solution stays clear blue.
Okay, Benedict's for reducing sugars. Check. So if that's for simpler sugars, how do we identify more complex carbs like starch, potatoes, bread, that kind of stuff?
Right, starch. It's a fascinating glucose polymer, and its unique structure is the key to its detection with Lugol's test. Unlike other glucose polymers, starch, specifically the component called amylose, forms a unique long-coiled chain, like a helix. A spiral staircase shape, sort of.
A helix. Okay.
This precise 3-dimensional coiling is exactly what the Lugol's test exploits. The elegant principle here is this. The iodine in Lugol's reagent, it's actually a potassium triiodide. Iodate gets physically trapped inside that starch helix. When the iodine molecules snuggle into that coil, they produce a distinct and unmistakable blue-black color.
Wow, so the iodine gets stuck in the coils.
Pretty much, yeah. And this is critical. Other glucose polymers, like cellulose, remember, plant cell walls, paper towels, they're also made of glucose, but they have a different uncoiled structure. So cellulose lacks this specific coiling, and it won't react with iodine to give that dark color. This makes the test highly specific for starch.
Specificity is key.
Definitely. The reagent is Lugol's iodine reagent, which starts as a yellowish-brown solution. A positive reaction with starch yields that characteristic, dramatic blue-black color. It really pops.
Blue-black means starch. What about the controls?
Good question. For controls for Lugol's, tube #3 gets a starch amylose solution that's positive. Tube #4 gets distilled water negative. Add a few drops of Lugol's reagent to both. Swirl gently. And that's it. Observe the color. No heating needed for this one.
Nice and simple.
So a positive Lugol's test turns blue-black. A negative Lugol's test stays that original yellowish-brown color of the reagent. And that paper towel example is great for visualizing this. Paper towels are 100% cellulose. Put a drop of iodine on one, it just stays brown. It really demonstrates the test is specific to the coiling of starch, not just glucose units.
That's a great illustration. Really drives home the structure function idea. Okay, now, on to proteins. The absolute workhorses. How do we test for them?
Right. Proteins, they're polymers of amino acids, but here's the nuance. The Biuret test doesn't target the individual amino acids themselves. Instead, it specifically targets the peptide bonds that link these amino acids together in the polypeptide chain.
Ah, the bonds between the amino acids.
Exactly. That selectivity is what makes it useful for detecting larger proteins or polypeptides. The principle is that the Biuret reagent contains copper 2 ions in an alkaline solution. These copper ions react with the nitrogen atoms involved in the peptide bonds. This forms a colored complex, specifically a characteristic violet color. Generally, the more peptide bonds present, meaning larger proteins or higher concentration, the more intense the violet color.
Copper again, but a different reaction, different color.
Yep. The reagent is Biuret reagent, which itself starts as a blue solution. A positive reaction produces that distinctive violet color. What it detects is proteins, or more precisely, any molecule containing 2 or more peptide bonds. This is important because a single amino acid, which has no peptide bonds, would not give a positive reaction. It needs that chain structure.
So free amino acids won't show up.
Correct. For the controls for Biuret, tube #5 gets an albumin solution. Egg white protein is common. That's your positive. Tube #6 gets distilled water negative. Add Biuret reagent to both. Swirl gently. And crucially, allow at least 2 minutes for the reaction. This color change can be a bit slower. Then observe.
Wait, 2 minutes. Got it.
So a positive Biuret test turns violet. Intensity varies with concentration. A negative Biuret test just stays blue, the color of the reagent itself. No protein detected. And again, remember a solution of just single amino acids, negative test. It really highlights that the test is looking for the polymer structure.
Makes sense. Okay, last one. Lipids, fats, oils, waxes, the water haters. How do we spot these guys?
Lipids, yes. A diverse group, but they all share that key trait. They are nonpolar and insoluble in water. The Sudan IV test cleverly leverages this. It works on that basic principle, like dissolves like.
Like dissolves like.
Okay. The principle is straightforward. Sudan V is itself a nonpolar dye. It's usually reddish. When you introduce this dye into a sample, if nonpolar lipids are present, the Sudan V dye will preferentially dissolve into those lipids. It seeks out its nonpolar buddies.
So the dye mixes with the fat.
Exactly. The reagent is a bit different. Usually a wooden applicator stick with Sudan V dye on one end, often the red end. The reaction and expected results are purely visual. If lipids are present, the red dye from the stick dissolves into the lipid. You'll see visible floating red droplets, or maybe even a distinct floating red layer if there's a lot of lipid.
Floating red droplets are a layer. Looks for fats and oils.
Yep, it detects lipids, generally fats, oils, etc. For the controls, 2 #7 gets corn oil, a good lipid, mixed with distilled water positive control. Tube #8 gets just distilled water negative control. Place a Sudan V dye stick into each tube. Stir vigorously and observe for several minutes. This one can also take a little time for the dye to really dissolve and become obvious.
Stir vigorously and be patient.
Right. A positive Sudan V test shows those visible floating red droplets or a distinct floating red layer. A negative Sudan V test, the red dye mostly stays on the stick or maybe disperses a tiny bit in the water, but you won't see those distinct red lipid structures forming. The dye just doesn't have a nonpolar place to go.
Okay, here's where it gets really interesting. Now that we've seen how to identify each molecule individually, we can apply these tests to real-world stuff, like food.
Exactly. Connecting this to the bigger picture, this is where you take everything, the tests, the controls, the principles, and apply it to figure out what biological molecules are in actual food items. Often in a lab setting, this is done in groups. Each person might test a different food, and then you pool your results. It's collaborative science.
Cool. So how does that work procedurally? Testing, say, a piece of apple versus some ground beef.
Good question. Let's walk through the general procedure overview for testing these unknown food samples. First, preparation. You'll get minced food samples. Mincing is key, increases surface area. Second, solubilization. Put about 3 scoops of minced food into 2 separate test tubes. Let's call them A and B. Add maybe 10 milliliters of distilled water to each and shake them vigorously. Really shake them. This helps pull out any water-soluble components into the liquid.
Get the soluble stuff out first, makes sense.
Right. Third, applying Benedict's and Biuret tests, using that liquid extract. From 2A, transfer about 2 milliliters of the liquid, try to avoid the solids, into a new tube, say tube C. Add Benedict's reagent, and remember...
Heat it. Boiling water bath.
You got it. Heat it, let it cool, then compare to your glucose and water controls. Patience again, sometimes the color takes time. Then, take another 2 milliliter of liquid from tube A into a different new tube, maybe tube D. Add Biuret reagent. Let it sit for at least 5 minutes. Compare to your albumin and water controls.
Okay, so Benedict's and Biuret use the water extract. What about Lugol's and Sudan IV?
Good question. Fourth step. Applying Lugol's and Sudan IV tests using the original food mixtures, tubes A and B, but with a crucial extra step. For starch, Lugol's, take tube A, the one with the minced food still in it. Place it directly into a boiling water bath for 5 minutes.
Wait, why boil the food mixture itself now?
Excellent question. Because starch often isn't very water soluble and it's locked inside plant cells with tough walls. Boiling helps rupture those cell walls, releasing the starch and making it accessible to the Lugol's region. You get a much stronger, more reliable result.
Ah, break down the cell walls. Smart.
Exactly. After boiling tube, remove it, add about 6 drops of Lugol solution directly into it, swirl, and compare to your starch and water controls. Similarly, for lipids, Sudan IV, take tube B, also with the minced food, put it in the boiling water bath for 5 minutes.
Same reason. Break open cells to get the lipids out.
Precisely. Fats are also not water-soluble and are often inside cells. Boiling helps rupture those cells and makes the lipids more accessible for the non-polar Sudan IV dye to dissolve into. After boiling tube B, remove it, put the Sudan IV dye stick directly into it, and stir vigorously. Again, observe for maybe up to 5 minutes, compared to your corn oil and water controls.
Boiling is key for starch and lipids in solid foods. Got it.
Absolutely. And throughout all this, there's some really important considerations. Number 1, preventing contamination. I cannot stress this enough. Use a separate, clean, disposable plastic pipette for every single solution or reagent you transfer. Seriously, mixing these up can completely wreck your results. You don't want your water control turning violet.
Right, 1 pipette per liquid. No double dipping.
Exactly. And second, patience and observation. As we've said, some reactions like Biuret and Sudan IV take time. Benedicts might need cooling. Don't rush your observations. Let the chemistry happen.
Good advice. Don't jump to conclusions. But this raises another question. What if things go wrong? What kind of tricky situations or unexpected results might pop up? Science isn't always neat.
That's very true. Critical thinking is essential. You might see results that seem unexpected, and figuring out why is a huge part of learning. One common issue is a false negative test. This is when the food does contain the molecule, but your test says it doesn't.
So the test misses it. Why would that happen?
Several reasons are possible. Maybe insufficient boiling for starch or fat. If you didn't boil long enough or hot enough, the molecule might still be locked in the cells, inaccessible to the reagent. Or maybe you didn't allow enough time for the reaction, especially for Biuret or Sudan IV. Read it too soon, looks negative. Improper mixing could be a culprit. If the reagent and sample aren't mixed well, no reaction. Or maybe the molecule is present but in such a low concentration dilution that the color change is too faint to see clearly. Looks negative, but it's just below the detection limit.
Okay, so several conventional pitfalls for false negatives.
Yeah.
What about confusing results? Like finding 2 things you didn't expect together.
Right, like understanding combined components. A great example is finding both starch and reducing sugar in something like a potato or a ripening fruit. Why might that be? It often reflects dynamic biological processes. During ripening, plants break down stored starch into simpler, sweeter reducing sugars using enzymes. So depending on the stage, how ripe the fruit is, or if the potato is starting to sprout, you can absolutely have both present at the same time, just in different amounts. It's not an error. It's biology happening.
Ah, so the unexpected result actually tells a story about the food's biological state.
Cool. Exactly. It requires interpretation, not just observation.
We've talked a lot about building molecules, dehydration, synthesis, but life also constantly breaks things down.
Yeah.
Right? Like when we digest food, what's the opposite process called?
That opposite process is hydrolysis. Literally means water splitting. Hydrolysis. It's the essential counterpart to dehydration synthesis, and it's absolutely fundamental. It's how our bodies digest the complex molecules in food.
Water splitting. So adding water breaks the bonds.
Precisely. During digestion, enzymes catalyze the breakdown of complex carbohydrates, proteins, lipids, and nucleic acids into their simpler monomers. This process involves the addition of water molecules across the bonds linking the monomers. That water molecule effectively breaks the covalent bond. The general equation looks like monomer plus HOO, monomer H plus monomer OH. You're basically putting back the H and OH that were removed during dehydration synthesis.
The reverse of building. Makes sense. How do we see this in the lab?
In this lab, you'll actually perform a carbohydrate hydrolysis experiment. The goal is to demonstrate this by breaking down sucrose, a disaccharide, and starch, a polysaccharide. You'll use heat and strong acid as catalysts instead of enzymes, but the principle is the same. You can't see the bonds breaking, but you can detect the products using our trusty Benedict's and Lugol's tests.
Ah, using the test to see the results of the breakdown. Clever. What products should we expect?
Good question. Let's think about the expected products of hydrolysis. When sucrose, remember it's glucose linked to fructose, is hydrolyzed, it breaks down into...
Fructose and fructose.
Exactly. And importantly, both glucose and fructose are reducing sugars. And when starch, A long chain of glucose units is hydrolyzed. It breaks down into...
Lots of glucose molecules.
You got it. And glucose, as we know, is a reducing sugar.
Okay, so hydrolysis creates reducing sugars from sucrose and starch. How's the experiment set up?
Right. The procedure overview involves a few stages. 1. Initial setup. Prepare several sets of test tubes containing your samples. A distilled water control, glucose control, sucrose, and starch. Let's call them A, B, C, D series. 2. Before hydrolysis analysis. This is your baseline. You'll perform the Benedict's test on the A series tubes, A1, A4, to check for reducing sugars before any treatment. Remember to heat these. You'll perform the Lugol's test on the B series tubes, B1, B4, to check for starch before treatment. Record these initial results.
Test everything first.
Got it.
3. Performing hydrolysis, C tubes. To the C series tubes, C1, C4, add your sample, some distilled water, and then add 1N hydrochloric acid, HCl. Big caution here. HCl is a strong acid. Handle it very carefully. It can burn.
Handle HCl with care. Check.
Gently swirl to mix and then heat these C tubes in a hot water bath for about 15 minutes. The heat and the acid together will catalyze the hydrolysis, breaking down the sucrose and starch. 4. Crucial step. Neutralization for Benedict's C tubes. After heating, remove the C tubes. Now this next bit is vital.
What? What's the catch?
The Benedict's test will not work correctly in highly acidic conditions. The acid interferes with the reaction, so you must neutralize the HCl first.
Ah, okay. How do you do that?
You'll add a small piece of litmus paper to each C tube. It should turn red, acidic. Then you add sodium hydroxide, NaOH, a strong base, drop by drop, swirling after each drop. Keep adding AOH until the litmus paper turns blue and stays blue. That means the acid is neutralized. Another swashin. NaOH is also caustic. Handle with care. Neutralize acid with base until litmus is blue. Careful with both. Got it.
5.
After hydrolysis analysis, now that the C tubes are neutralized, you perform the Benedict's test on them, C1, C4. Remember to heat. For the Lugol's test after hydrolysis, the procedure usually involves taking a sample from the hydrolyzed C tubes, maybe transferred to D tubes, and adding Lugol's. Neutralization isn't as critical for Lugol's as it is for Benedict's.
Okay. Test the hydrolyzed samples with Benedict's after neutralizing. And Lugol's. What should we see?
This is where you see the proofs. Let's interpret the results. For sucrose, before hydrolysis, Benedict's should be negative. It's non-reducing. Lugol should be negative. It's not starch. After hydrolysis and neutralization, Benedict's should be positive because you make glucose plus fructose, both reducing. Lugol's should still be negative. That change in Benedict's proves sucrose broke down. Negative Benedict's becomes positive. Cool.
Yep.
What about starch?
For starch, before hydrolysis, Lugol's should be positive. Blue-black, it is starch. Benedict's should be negative. Starch isn't reducing. After hydrolysis, Lugol's should become negative. The starch is gone. And Benedict's should become positive because you broke the starch into glucose, which is reducing.
Positive Lugol's becomes negative. Negative Benedict's becomes positive. Very clear changes.
Exactly. Those changes in the test results before and after hydrolysis provide clear evidence that the complex carbohydrates were broken down into simpler sugars.
So what does this all mean? We're literally seeing complex molecules getting chopped up into their simpler, sweeter components, just using acid and heat. It's a fundamental process our bodies do all the time with enzymes. Amazing to witness it, even indirectly, through these tests. Okay, we have covered a ton of ground in this deep dive. Wow. From the basic building blocks, the monomers and macromolecules, to the specific tests like Benedict's, Lugol's, Biuret, Sudan IV, the importance of controls, testing actual food, dealing with potential issues like false negatives, and even breaking molecules down with hydrolysis. Now, for the really practical bit, how does all this help with those pre-lab quizzes?
Right, the pre-lab quiz. Which leads us to wonder, what are they actually for? They're more than just a quick grade grab, They're a really vital part of your learning process, getting you ready for the actual science.
How so?
Well, their purpose is really threefold. First, they force you to focus on the key learning objectives before you start messing with test tubes. What are you supposed to learn today? Second, they stimulate scientific thinking. Get your brain in gear before the experiment. Anticipate results. Think about procedures. And third, they help you spot any confusion or gaps in your understanding early so you can ask questions before you make a mistake in the lab itself.
So like a mental warm-up and a safety check.
Pretty much, yeah. In terms of format and importance, usually 2 short questions, maybe 1 point each. Quick, about 5 minutes. But crucially, they add up. They contribute to your final lab exam grade, often around 10% of the semester's lab grade. That's significant. And the kicker, They're given right at the start of class if you're late. You get a 0 for that quiz, no exceptions usually. Punctuality is key.
Ouch. Don't
Yeah.
be late for lab. Okay, what about that cheat sheet strategy?
Ah, yes, the index card. Often you'll get an index card to make notes on beforehand. You fill it out by hand with key info, tips, procedures, results. The act of physically writing it down really helps cement the information in your brain. It's way better than just reading. Then you turn the card in with your quiz.
A sanctioned cheat sheet you make yourself.
Oh, nice.
So based on our deep dive today, what absolutely needs to go on that index card for Lab 3? What kind of questions might pop up? Give us the inside scoop.
Okay, let's brainstorm some potential questions and what you'd jot down. Remember, concise notes are key for the card. Potential question 1, conceptual. Explain the importance of a negative control in the Biuret test and what result you would expect from it. Card note. Neg control, DW, shows no contamination region issue. Biuret plus DW stays BLE, no protein, no violet.
Okay, short and sweet. What's another one?
Potential question 2, procedure principle. Describe the principle behind the Benedict's test for reducing sugars, including the expected color change for positive result. Card note, Benedict's. Reducing sugar, CO, reduces QU, blue, PPU. Oh. Pose, blue, green, yellow, orange, or lambda, PPT.
Good one. How about variables? They often ask about those.
Right. Potential question 3, application variables. If testing unknown food for fat with Sudan IV, identify the independent and dependent variables. Card note. Sudan IV, 4V, presence, absence of fat in sample. DV, appearance of red droplets layer. Yes or no?
IV and DV. Classic. What about hydrolysis?
Definitely. Potential question 4, hydrolysis. Define hydrolysis and give a lab example using carbohydrates, noting the test result change. Card note. Hydrolysis, break bonds using HUO, X sucrose plus HUO, acetate, glucose plus fructose, Benedict's, negative before, positive after.
Excellent examples. What other key info should be crammed onto that card?
Okay, other key info for the cheat sheet. Monomers, quick list. Carbs and monosaccharides, proteins, amino acids, lipids and glycerol fatty acids, test regions and positive results, maybe a quick table. Benedict's, blue, Biuret PPT, red sugars, heat, Lugols, brown O, blue black, starch. Biuret, blue violet, protein, Sudan IV. Red dye, red droplets layer, lipid. Crucial procedural notes. Why heat Benedict's? Reaction needs it. Why boil food for Lugol Sudan IV? Rupture cells. Why neutralize after hydrolysis for Benedict's? Acid interferes. Use separate pipettes. Contamination. Key distinctions. Reducing versus non-reducing. Sucrose non-reducing until hydrolyzed.
Wow, that's a packed index card, but super useful. That should definitely help anyone nail the pre-lab quiz.
Having that concise handwritten summary is really the best way to walk in feeling prepared and confident. It's about understanding the why, not just the what.
Absolutely. Wow, what an incredible journey into the heart of biological molecules today. From their basic structures, the monomers and polymers, to the clever tests we use, Benedict's, Lugol's, Biuret, Sudan IV, understanding controls, testing food, even hydrolysis. Ooh, we really covered it all.
We really did. And, understanding these core concepts and the meticulous procedures isn't just about passing a lab. It genuinely helps us appreciate the complexity of the food we eat, the amazing chemical process happening inside us constantly, and just the sheer elegance of biological chemistry.
So what does this all mean for you, our listener? Well, next time you look at an apple or some pasta, or think about how your body digests lunch, you'll have a much deeper, more informed appreciation for that hidden molecular world at play. This knowledge isn't just for the lab, it's for life. It changes how you see the world.
It really does. And maybe a final provocative thought to leave you with. If our bodies are constantly performing these incredibly precise hydrolysis and dehydration synthesis reactions, balancing everything perfectly to keep us alive and provide energy, how might even a subtle disruption in these delicate chemical processes, maybe from diet, genetics, environment, impact our overall health and well-being? What could be the real-world consequences of these tiny, microscopic changes rippling outwards? It really makes you think about the intricate dance of molecules that is life itself.